Hi, so good morning, good evening, everyone. Welcome to Singapore virtually. So today we're really excited to have two talks. The first talk is going to be by Dr. Anthony Khong. So he's a new principal investigator at the Cancer Science Institute of Singapore and National University of Singapore. He earned his PhD in biochem and molecular biology at University of British Columbia with Professor Eric Yan, studying viral host interactions. In 2016, he joined Roy Parker's lab at UC Boulder, and he's been working on stress granules. And one of his most exciting work is his work which suggested that RNA recruitment to stress granules is not specific. And this was in contrary to certain other beliefs in the field that RNAs are actively sorted into stress granules for differential gene expression. So this has also been supported by other work from the Parker lab and has led to the hypothesis that stress granules are RNA aggregates which are driven by non-specific RNA-RNA interactions. So currently, Anthony's work is centered on investigating functional aspects of these RNA aggregates in cancer biology, and we're really excited to hear his talk today. So over to you, Anthony.
Thank you for the introduction, and I also want to thank the organizers for this amazing opportunity to present my work today. So let me first share you my screen, and I'm going to stop my video in case the video cuts off the screen. So I stop the video. There you go. All right. So what I want to tell you today is a new function of stress grounds, promoting cell cycle exit and chemoresistance. So before I get into stress grounds, let me first tell you about what are stress grounds. Stress grounds are a type of cytosolic messenger ribonucleic protein granule. They're formed during cellular stress, hence the name stress granule. These stresses can be oxidative stress, heat shock, hypoxia, virus infection. What you really need to know is that during cellular stress, this leads to an integrated stress response, and ribosomes are run off, and you get this accumulation of non-translating messenger ribonucleic proteins. And this leads to the coalescence of these MRPs, and this leads you to forming stress granules. Stress granules are shown right here. The green is a protein marker for stress granules, and red is a particular mRNA that found to be enriched with stress granules. Right, so why should you care about stress granules? Stress granules are thought to be involved in RNA translation control because it's often linked to regulation of gene translation. Moreover, it's been shown to be linked to neurodegeneration in cancer biology. Shown here is a pancreatic ductal adenocarcinoma from a patient, and you see a lot of stress granules. And moreover, it's also been used to study a fundamental aspect of cellular organization. And I'm sure you've heard of this term biomolecular condensates, so it's often used as a model to study biomolecular condensates. So what we really would like to know is how do stress granules assemble, what are their composition, and what are their functions? So back in 2016, when I joined Roy Parker's lab, I wanted to ask some very basic questions about stress granules. And most in particular, I wanted to ask what RNAs are sorted into stress granules. And there was this prevailing model in the field that some RNAs are selectively sorted to stress granules, and that conferred differential gene regulation. For example, you can imagine perhaps particular RNA sorted into stress granules, and that allows it to be more stable, so it resists degradation. So the primary goal is to examine how stress granules modulate gene expression by sequestering specific RNAs. And my secondary goal is to examine how mRNA contributes to the stress granule assembly. And the approach I took was biochemically isolate stress granules and analyze its transcriptome. And so just to give you the punchline, what we discovered is actually quite surprising. A non-specific RNA aggregation is actually mediating the formation of stress granules. In fact, bulk RNA accumulates in a relatively non-specific manner that's based on relatively two simple metrics: release from ribosome and RNA length. So what we realized that rather than thinking RNA in general is sorted into stress granules, RNA is actually very much involved in the process of creating granules by non-specific RNA interaction. And this leads to the idea that perhaps stress granules are RNA aggregates. And so I would point you to a very nice review written by my friend Nina Rippin on this very controversial idea, I would say, about maybe stress granules actually should be thought of as RNA aggregates. And this model was done by many people in Parker Lab that I have tremendous opportunity to work with over the past 6 years. This includes Tyler Matheny, Brina Van Treek, Stephanie Moon, David Telbier and Gabriel Telbier, Nina Repin, and of course, Roy Parker, and many others in Parker Lab. So this begs the question then, if stress granules are RNA aggregates, are they actually functionless then? So I think cells are pretty clever. They would have figured out how to use this bug of physical properties that RNA tends to aggregate in order to confer some kind of function. Just like humans who have taken advantage of water and gravity to generate electricity. So then I imagined, okay, where might stress granules actually play an important role in biology? So I thought perhaps cells that encounter a lot of different extrinsic stressors like hypoxia, oxidative stress, ER stress, or cells that tend to overexpress many of the components of stress granules. And I would like to suggest today that cancer biology is actually a nice area to look at for stress granules and perhaps address whether stress granules actually have functions. So there are a couple of reasons to look at this in cancer biology. First, it's already been noticed that stress granules are seen in a number of different patient cancer cells and tumors. Recently presented RNA-CSI meeting 2022, where it's been seen in patients from acute myeloid leukemia. It's also been seen in medulloblastoma, pancreatic ductal carcinoma, and sarcoma. I also want to mention that stress granule protein expression often correlates with poor patient prognosis. In fact, high expression of the scaffolds are responsible for making stress granules, like GP1, 2, and TI1 caprid, are often linked to poor patient outcome. Conversely, expression of a stress granule negative regulator, such as USB10, often lead to better patient outcomes. And it's been proposed that stress granules are actually implicated in many different cancer processes, ranging from transformation to proliferation to stress resistance, all the way to metastasis. But I just want to emphasize molecular mechanisms are not really well understood. So one area of biology that's been studied quite a bit between stress granules and cancer biology is chemoresistance. For example, it's been shown that stress granules form in a variety of chemotherapeutics. Shown here is a vinca alkaloid where I've treated cells for 1 hour to induce stress granules as spironolactone. It's also been known that inhibiting stress granule formation can sensitize cells to chemotherapeutic cell death. For example, in U2S cells where I've treated spironolactone for 3 hours on wild-type cells, I see many more cells surviving compared to a stress granule deficient cell line. So the major question in the field is how are stress granules promoting chemoresistance? And of course, there are many different mechanisms that cells can use to promote chemoresistance, including increased DNA repair mechanisms, inhibition of apoptosis, increased drug influx, and promoting the slow-cycling quiescence state. And I'd like to suggest today that perhaps stress granules are promoting the slow-cycling quiescence state in order to confer chemoresistance. So the inspiration behind this work stems from work that was being done in a neighboring lab at CU Boulder from Sabrina Spencer's lab. And they've noticed that a number of different cell lines actually can engage in the slow cycling quiescence state, specifically minority cell lines. And I thought, okay, if these cells are engaging in the slow cycling quiescence state, how did they begin that way? So they realized by lineage tracking, they start off as a stress cell, meaning they were engaged in integrated stress response. So I thought, okay, if it's engaged in integrated stress response, is it then promoting stress granules? And are these stress granules then promoting the slow cycling quiescence state? So I decided to look at this and made some predictions. So the first prediction I made was cells with stress granules are likely non-proliferating then. So in order to study this, I took a cell line that was stably expressing a stress granule marker, GP1-GFP, and staining with phospho-RB, and asked for cells that have stress granules, do they lack phospho-RB staining? Phospho-RB is a marker for proliferation. And since this is done under non-stress conditions, I had to image a ton of cells to find stress granules. So I used a high contact screening microscope, the Opera Phoenix, to find these cells that have stress granules. And so I discovered that, in fact, sporadic stress granule formation often correlates with non-proliferating state. Shown here is a representative example of that, where these two cells have stress granules and they lack phospho-RB staining. And I've done N=3 biological replicates, and they all seem to correlate with non-proliferating state when cells have stress granules. And I see similar results with also wild-type U2S cells. So this suggests stress granules are involved in promoting cell cycle exit, but this is a correlation. So to address this, I decided to ask whether wild-type cells have more non-proliferating cells than a G312 double knockout cells, which is a stress granule deficient cell line. So I see, in fact, that wild-type cells have more non-proliferating cells than G3 double knockout cells. And shown here is an image to represent that, where I see some fraction of cells that have low phospho-Rb staining compared to a stress granule deficient cell line. And this can be rescued with a G3B1 GFP gene. And shown on the right is a quantification to support this. So this suggests stress granules are indeed promoting cell cycle exit. So then I asked, okay, if it's true under non-stress conditions, can I elicit more cell cycle exit state with a stress condition? And do I see more non-proliferating cells in the wild-type cells compared to a stress granule deficient cell line? And I actually see that. So in wild-type cells, I see many more cells that are non-proliferating now compared to the G3B12 double knockout cells. So this suggests that stress granules are promoting cell cycle exit for stress. But there's actually an issue. And the issue is G3B have actually many other functions besides assembling stress granules. It's well known that G3B is involved in C-gas signaling in a variety of RNA metabolism processes such as RNA decay, translation, ribosome degradation. So it's really hard to draw the connection between G3B knockout and a stress granule function. So it's been a major challenge in the stress granule field, and also from the biomolecular cognoscience field, I would say. A G knockout doesn't necessarily mean a stress granule knockout. So to address this, I've taken two different approaches to rescue stress granules in the G3B double knockout cells, and to ask if they form similar levels of non-proliferating cells. So there's two approaches I talk. One is a chemical approach, and one is a genetic approach. So it's been known that with a chemical approach, they can elicit stress granule formation using this thing called sorbitol, and this can rescue stress granules in the G312 double knockout cells. So I thought, can sorbitol then rescue the level of non-proliferating cells? And so that's actually what I see. So by imaging, these are represent the examples where I see low phospho-Rb staining for cells in the wild-type cell line, and now also in the G3 double knockout cells. And I see similar levels by quantification on the right. So these results suggest non-proliferating cells is dependent on stress granules and not simply dependent on the G312 genes. So then I took a genetic approach. So I asked, can I rescue stress granules with a synthetic gene that can then promote cell cycle exit? So this was excellent work done by Paul Taylor's lab, where they showed that they can replace G3B, but kept the multivalency and showed that I can rescue stress granules. How did they do this? So they replaced the NTF2L domain, which is found in G3B, which allows G3B to dimerize, with a GST domain, which allows this artificial protein to dimerize. Then they also replaced RNA domain with another RNA from HRPA1, and they replaced the IDR domains. So this is a completely different protein, and yet it can still rescue stress granules. So that I use the system and show, hey, does it rescue also this non-proliferating state? And that's actually what I've seen. So when I look to stain for phospho-Rb, I see there's similar levels of cell cycle exit in wild-type cells and the GFP synthetic cells with the G3B12 double knockout compared to the G3B12 double knockout. And I want to mention this done under stress conditions, but also looked under non-stress conditions. I see very similar results too. So these results suggest non-proliferating cells is dependent on stress granules and not just dependent on the G312 genes. So just to summarize, what we have now is a model that suggests that stress granules are promoting cell cycle exit, which then in turn promotes chemoresistance. So this raises two important questions. What the molecular basis that explains how stress granules promote cell cycle exit, and is it truly promoting chemoresistance? So just because this fluorescence is very well studied, we thought, hey, is there a particular protein that is well known to be involved in promoting cell cycle exit? And one of these is P21. So we decided to investigate if stress granules are promoting P21 levels. So we actually see that. So we found that in wild-type cells, we see much higher levels of cells that contain P21 proteins compared to the G3B12 double knockout cells. And this can be rescued with a GFP synthetic. And shown on the right is this quantification for that. And the results are also consistent. It's done under stress conditions, but also consistent under non-stress conditions. And I also want to mention that P21 expression is rescued sorbitol in the Judy-Bolden double knockout cells. So another means to show that it's stress granules, not just G3P. So this provides an explanation for how perhaps stress granules may be promoting cell cycle exit. The second important question I wanted to ask is, are chemo-induced stress granules actually promoting cell cycle exit and chemoresistance? So going back to that drug that I used earlier, this being alkaloid, I decided to ask if this actually induces cell cycle exit and promoting chemoresistance. So shown here is the results. I do see stress granules promote cell cycle exit and when cells are treated with melamine. And shown on the left is images supporting this. So in the wild-type cells, I see many more cells that have low phospho-RB staining and compared to the GTP1/2 double knockout cells. And shown on the right is quantification. So then I asked, are stress granules promoting granulomatous resistance? So to do this, I stress U2S cells with 3-hour melamine, wash with PBS, and trypsinize and seed the same number of cells, and then perform colony forming assay. I found many more cells in the wild-type cell line forming colonies compared to the stress granule deficient cell line. And as I mentioned, the results are consistent with 2 other biological replicates. So these results suggest stress granules not only limit cell death, but also allows the cell to recover and proliferate again, which is rare reminiscence of a quiescence phenotype. So to summarize, what I've told you today is chemotherapy can induce stress granules, and these stress granules can promote this non-proliferative state, which allows it to survive the chemotherapy, and then perhaps re-enter the cell cycle and then repopulate a tumor. And so this suggests that inhibiting stress granules could be an advantageous therapy for chemotherapy. So perhaps combining chemotherapy and stress granule inhibitors, you may be more efficient at killing the tumors. The other point I want to mention is quiescence and non-proliferative state may actually provide an explanation for other stress granule functions. For example, stress granules known to inhibit apoptosis, reduce reactive oxygen species, promote metastasis. And perhaps stress granules operating through these mechanisms by promoting quiescence, because quiescence is also known to inhibit apoptosis, reduce reactive oxygen species, and promote metastasis. So lastly, I just want to tell you about my new lab. That I have a few goals to aim for. First, what is the molecular mechanisms underlying the regulation gene expression by stress granules, including P21? Identify the types of tumors in which stress granules are present, and develop cancer models incorporating disease-relevant stress granules and investigate their role in tumor biology. If you're interested, please contact me. There's opportunities for graduate student research in the laboratory now. And I also want to mention that CSI is a program to recruit graduate students. There's a competitive stipend that's offered, so feel free to take a snapshot of this QR code if you're interested. And finally, I'd like to thank my previous lab, Roy Parker, who's been instrumental in shaping my career, Nina, Louisa, who contributed to this latest work, discussion from many people at CU Boulder, Oki Lindbach, William Spencer, Victor Passanini, the rest of the Parker Lab, reagents provided by Nancy Kurdash and Paul Anderson, the Biofrontiers core facilities. I'd like to thank previous funding sources, HHMI, NIH, and Advancing Constructive Fellowships. I'd like to thank all of your attention. So I'd like to open up for questions.
Thank you, Anthony. That was a really interesting talk. There's one question in the chat, but I think it's more for the organizers about whether there's closed captioning or not. Yeah. There's a question about, did you check the vincristine expression level after transfection?
Maybe you can rephrase that question, Psyche. I think what you imply is, did I check the G3P expression after adding chemotherapy?
I'm not sure. Does the ZKA, does that answer your question? Okay, while we're waiting, there's a second question from Stephen Rader about whether the link between stress granules and quiescence is conserved, for example, in yeast.
Hi, Stephen. We haven't looked in yeast to see if there's a correlation there. So that's an exciting thing to perhaps look at.
There's another question about whether you explored isotope labeling of the bases.
I'm not sure how to answer the question, but thank you for the question.
Maybe I'll ask you other questions next week, but a quick question, because you mentioned G3BP1 has got multiple functions in different RNA processing steps. So how do you for sure attribute that the knockout phenotype is due to just the loss of stress granules?
Yeah, so it's very difficult to just attribute to stress granules. So that's why I did those two different approaches, where I try to rescue stress granules, we knock out the chemical approach, and also using this GFP synthetic method. So I think, let me just stop sharing. I think with those two approaches, we can probably argue that is due to stress granules, but I wouldn't say I'm like 100% confident until we figure out actual molecular mechanism. So until we figure that out, then I think we're much more confident that it is component cell structure.
Do you see any specific non-coding RNA species linked with the stress granules? Because you know a lot of like the speckles have defined like non-coding RNAs.
Yeah, so this was published back in my 27th study, where we do see a bunch of linked RNAs in stress granules, and one of this is NORAD, which is this really long non-coding RNA that's involved with genomic stability, and it's highly enriched in stress granules. About 80% or 90% of molecules are in stress granules. Whether that has any role in regulating NORAD's function, I'm not sure.
Okay, sorry, Anthony. You can ignore my question. Now you have 3 more questions, and then I think we have to stop. Is it known why G3BP1 and 2 aren't required for stress granule formation in the context of sorbitol stress?
The mechanism isn't known, but if you allow me to speculate, I think it's promoting more RNA interactions. So in previous work, where we showed we can rescue stress granules in the absence of G3BP1/2 proteins by inhibiting EI4A. And so that would lead to more transient RNA interactions and rescue stress granules. So I think sorbitol may be working through that manner.
The next question about whether there's any live staining markers that are not GFP fusion.
That's a great question. I haven't done any of that. I haven't seen any literature that's, I've tried a few things out there. It hasn't really worked in my hands of live staining markers of stress granules. That's not GFP fusion protein. Or you mean any, so people have done DS-RED, other fusion, fluorescent fusion proteins. But you've mentioned like a small dye that binds to stress granules. I haven't seen anything that works. At least in my hands.
Okay, this is a really interesting question from Lisa Hall about is the non-specific RNA aggregation made in order to sequester specific proteins instead of to sequester RNA?
That's a really good question. I don't have an answer to that right now. Possibly. So I don't have an answer to that. Thank you for the question.
Okay, so I think we're good. Thank you so much again, Anthony. And then I think we can move on to the second talk for today.
Thank you.
Okay, so our second speaker for today is Dr. Pek Jun Wei. So Jun Wei did his graduate research with Dr. Toshie Kai at the Temasek Life Sciences Laboratory and the National University of Singapore. He studied the role of small RNAs in Drosophila germline. And then he did postdoc training in the labs of Dr. Joseph Gall and Alan Spratling at Carnegie Institute for Science. And he was a Carnegie Collaborative Fellow as well as HHMI Fellow of the Life Science Research Foundation and a Jane Coffin Childs Fellowship as well. So he returned to Singapore in 2014. He started his independent research. He's made many exciting discoveries about a novel class of non-coding RNAs, which are called stable intronic sequence RNAs or cis-RNAs. And he's shown that these cis-RNAs engage in regulatory feedback loops, and they play an important role in stem cell homeostasis and development. And he's currently extending his research to cancer. So really excited to hear about his work today. Jun Wei, over to you.
Thank you so much, Yvonne, for the very kind introduction. Let me share my slide. All right. Hi, everyone. Thank you. Thank you again for the introduction and the opportunity to share our work in this very exciting seminar. So let me. Okay, so my lab actually, the work of my lab actually focus on a specific so-called class or species of non-coding RNAs that are coming from the introns. So before I start, I just want to bring you all to a very basic gene expression program that I believe every one of you know what. So in the eukaryotic cells, most of us know about that gene expression starts from the transcription all the way to translation. But I think many of us are very protein-centric or maybe non-coding RNA-centric, but many times we do not really appreciate what happens to the introns after they are being spliced out. So during splicing, besides that messenger, the exons are being joined together, the introns are being spliced out in the form of layers, and this layer is then rapidly degraded in the nucleus. However, if you look at the amount of genetic material that are present in the genetic material that's if you look at the amount of genetic materials that are present in the genome, the amount of genetic material that encodes for the intron is far more than the exons. So in a typical human gene, the amount of introns is actually even 10 times more than that of the exons. And if you compare the number of introns from a basic bacteria all the way up to a very complex human species like us, we can appreciate that the number of introns actually increases over the developmental complexity. However, in the past, or maybe only until recent years, we start to actually, we still do not really think that introns actually have some functional role in the cells. The main reason is that people usually think that introns are used as byproducts of splicing. And one of the main reasons is that they are extremely unstable, as I mentioned before, and they are degraded in the nucleus in a matter of seconds or minutes. Another area that why people don't think that introns have any function is the lack of sequence conservation. However, we know that there are certain exceptions. For example, we know scar RNA and SNO RNA and some microRNAs actually are coming from the introns. However, these are only the tip of the iceberg. In the past 30 years, if you look at this slide, we realize that there are already some studies back 30 years ago that talked about certain stable introns that are present in viruses, in Drosophila, in mammalian cells, and even some vertebrates such as a frog. However, at the point of time, people usually think that these are exceptions rather than the norm. So the story started actually while I was a postdoc in Joe Gall's lab in Carnegie Institution. So Joe is very interested in understanding what are the genes that are expressed during the frog oogenesis. So as you may know, the frog oocyte is a very huge cell, whereby he can actually successfully dissect out the nucleus or germinal vesicles using a pair of forceps. And cleverly, he devised some methods that enables him to remove the nuclear envelope from this nucleus in order to get pure RNA fractions from the nucleus. So he sent this this nucleus for sequencing, and what he found was totally unexpected at that time. In the cytoplasm, as you can see, we can appreciate that there are spliced reads, exons, reads that are mapped to the exons. However, in the nucleus, he was expecting to see pre-mRNA or nascent transcript. However, what he saw was that he saw abundant intronic reads that map to they're present in the nucleus. These are actually not nascent transcript because there are very, very little reads that map at the intron-exon junctions. So this is actually one of the first observation that such intronic reads are present in the nucleus for more than 90% of the genes that are expressed during the point of time. And further experiments actually show that these introns are unusually stable for at least 2 days, as compared to what I told you in a matter of minutes or seconds. So these introns are extremely or unusually stable. So with this observation, he coined this phenomenon stable intronic sequence RNA, or in short, cis-RNA. So you can appreciate that the timeline of cis-RNA discovery actually took around 20 years from the early exceptions to now that we realize that these are not exceptions anymore due to the improvement in the technologies that we have. So now, stable introns are actually not exceptions, but a general phenomenon that we see in many, many different model organisms that we have looked at so far. And in my lab, we have actually used Drosophila to study the functions of these cis-RNAs, in particularly looking at how they regulate different stages of oogenesis, right from the stem cells to the maternal pool of cis-RNAs. So for example, in the stem cells, we have characterized 2 different cis-RNA that regulate stem cell homeostasis. And we also characterize a number of maternally inherited cis-RNA that actually help the mothers actually regulate the gene expression of their offspring. So our studies and others have found that besides generating cis-RNAs from the canonical splicing events, such as what I've shown here, cis-RNAs can also be generated by other types of splicing events, such as back splicing, whereby part of the intron is being retained in a circular molecule. Also, some of the cis-RNAs can be generated by normal transcription, whereby these introns are being retained in the final transcripts. And just to take note that these transcripts are usually stable, that's why we call them cis-RNAs. So the purpose of this talk, I just want to introduce one type of cis-RNA that I will show you today that is formed by this intron retention event. So during that point of time, maybe 5 years ago, my postdoc, Chris Chan, actually entered the lab. He's actually have some background in studying human cells, although my lab focuses on Drosophila cells. But with his expertise in human cells, he would like to actually try to find whether there are cis-RNAs that are formed by intron retention in human cells and what are their possible functions in regulating gene expressions. So we decided to actually look at this gene called AGLU1. So AGLU1 has been characterized previously by Gordon Carmichael lab, whereby he has shown that AGLU1 can be spliced into different ways. So one of the ways is canonical splicing, which produces the canonical AGLU1 messenger RNA. And the intron 2 of AGLU1 can be alternatively spliced to retain this yellow poison exon, which leads to the formation of an unstable transcript, which is degraded by NMD. And in his paper, he also showed that this intron 2 can also be fully retained in the final transcript as shown here. And this transcript seems to be stable and is retained in the nucleus as well. One of the interesting things about this intron 2 is that it contains a stretch of ultra-conserved element, or we call UCE, that actually spans this poison exon. So we thought that it would be interesting to further characterize this potential cis-RNA. So the first thing that we have done is that we look at the stability of these 3 different transcripts by using alpha-amanitin treatment. So I hope that you can appreciate that actually the messenger RNA is really very stable, and the cis-RNA is relatively more stable than the pre-mRNA as shown here. And when Chris did the single molecule FISH to detect for the localization of this cis-RNA, he found that this cis-RNA is exactly localized in the nucleus as shown here. And some of the this transcript actually colocalized with the pre-mRNA, which shows that actually this transcript could be nascent transcripts. So by quantification, we found that the AGLO1/CIS RNA is far more abundant than the pre-mRNA, which is consistent with it being more stable than the pre-mRNA. And he also further confirmed this by doing nuclear cytoplasmic fractionation and showed that this AGLO1/CIS RNA is indeed present in the nucleus. So we are interested to look at whether this UCE is important for the formation of these cis RNAs. So he actually designed 3 different AMOs that block this region of the UCE, and he found that after transfecting this AMO into the cell, the biogenesis of the cis RNA is diminished, as shown here. So it seems like this UCE is important for the biogenesis of the cis RNAs. So next, he's interested to look at what is the potential function of these cis RNAs. So he used 2 different antisense oligo, or ASO, to target the intron 2 of these cis RNAs. So the knockdown is efficient, and at the levels of the ASO that we are using, we do not see that this ASO to be targeting the pre-mRNA. So this ASO is specific to the cis RNA, but not the pre-mRNA. And in this context, he found that after he knocked down the cis RNA, the splicing index of this intron 2 actually decreases, suggesting that this cis RNA actually promotes splicing of the intron 2. So he did the reciprocal experiment whereby he tried to overexpress this intron 2 to see whether he can see the opposite effect. So the answer is yes. When he overexpressed this intron 2, he could show that the splicing index actually increases. Interestingly, if he mutate or remove this UCE region, this effect is diminished or abolished. And this is associated with a reciprocal change in the cis RNA levels. With higher splicing index, the cis RNA expression or biogenesis is decreased. So the next question is, how do these cis RNA regulate splicing? So we know that actually from previous study, the AGLO1 actually can bind to the AGLO1 pre-mRNA, and binding of this actually can repress splicing of this intron 2. So we hypothesized that this cis RNA could be actually binding to this AGLO1 protein, whereby it is regulating its activity. So the first thing we check is whether this AGLO1 can actually bind to AGLO1 protein by pulling down AGLO1 protein. And indeed, when we pull down AGLO1 protein, we find an enrichment of this cis RNA, indicating that AGLO1 protein actually binds to the cis RNA in the complex. When we knock down this cis RNA, we find that AGLO1 binding to cis RNA decreases, as shown here, and this actually leads to a higher binding of the AGLO1 protein to the pre-mRNA, indicating that when there is little cis RNA left, the AGLO1 protein can now move on to bind to the pre-mRNA and then inhibit splicing. So next, we actually look at where does this cis RNA localize in the nucleus? What could be the potential molecular function? So we found that actually AGLO1 protein and cis RNA actually co-localize in nuclear bodies as shown here. And the AGLO1 protein has been previously shown to be localized to the nuclear speckles as well. So we double-check this by co-staining AGLO1 protein with SC35, the nuclear speckle markers, and we found that they nicely co-localize with each other. So this data actually shows that AGLO1/CIS RNA localized to the nuclear speckles. So the localization of the AGLO1 protein to the nuclear speckles is actually quite dynamic. We perform FRET experiments whereby we photobleach one of these nuclear speckles that contain AGLO1 protein, and we found that after photobleaching, the signal of AGLO1 protein actually recovers pretty quickly, comparable to another nuclear speckle protein, SRSF2. So the localization of this AGLO1 protein to the nuclear speckles is quite dynamic. And next, we try to see whether is this cis RNA important for the localization of AGLO1 protein to the nuclear speckles. So in control cells, we see AGLO1 protein co-localized with SC35 as shown here, nicely in the nuclear speckles. However, if we knock down agglutinin RNA, we don't see any changes in the localization of the SC35 nuclear speckles, indicating that nuclear speckles formation is perfectly fine. However, we see that now agglutinin protein do not localize to nuclear speckles very well, as shown by the diffuse state of the agglutinin staining. So it seems that agglutinin RNA is important for robust localization of the agglutinin protein to the nuclear speckles. So what is the potential molecular mechanism? So first, I think we check whether this agglutinin protein can indeed form condensates in vitro, because many of these proteins that localize to nuclear speckles can form biomolecular condensates. So in vitro, we found that actually agglutinin actually can form condensates, and these bodies actually increase in increasing amount of agglutinin protein. And what happens is when we add agglutinin cis-RNA together with this in vitro RNA, in vitro agglutinin protein, we found that if we add agglutinin cis-RNA, these condensates become bigger, and they are actually form more condensates as shown here. So the number of droplet actually increases when we add in intron 2 of the agglutinin cis-RNA, and those condensates that contain this agglutinin cis-RNA actually are also bigger than those condensates that do not contain the agglutinin cis-RNAs. And as a control, we look at what happens if we add this agglutinin cis-RNA without the UCE. And without this UCE, it loses all its activity in producing this condensate formation. So agglutinin cis-RNA binds to agglutinin protein, and this requires a UCE for its activity. So with this, actually, we came up with a model whereby the production of this agglutinin cis-RNA actually is in a temporal process, and this is linked to its downstream function. So we hypothesize that the agglutinin protein usually will bind to the intron 2 of the agglutinin pre-mRNA, and this leads to intron retention. And intron retention eventually leads to cis-RNA formation as shown here. So the formation of this cis-RNA then in turn feedback and pull this agglutinin protein to the nuclear speckles, and this again induce localization of more agglutinin protein to the nuclear speckles, whereby inhibiting the activity of agglutinin protein. So we think that this is a way of how this system actually achieve homeostasis in gene expression. So interestingly, because our lab also are interested in looking at flies, so we actually found that the fly agglutinin gene actually has the same exon-intron structure as the human one. It has actually 4 different exons and 3 different introns. And interestingly, we found that the intron 2 of the fly agglutinin also produces cis-RNAs. However, this cis-RNA is produced in a different way. Instead of retaining the entire intron 2, the cis-RNA actually is produced by premature cleavage of the intron 2. So because of this, we wondered whether if we put this human cis-RNA back into the fly cell, how will this human cis-RNA being processed? So actually what Chris did was that he made a mini gene with the exons and then agglutinin intron 2 and transfect it in Drosophila S2 cells. So what happened is that after transfection, he did not see that the production of the agglutinin cis-RNA, as this intron 2 is not being retained as what we have seen in the human cells. Instead, the intron 2 is being spliced into normal messenger RNA and also into many, many different types of alternative splice transcript that can be potentially degraded by NMD. Associated with this, we also do not see that any effect of transfecting intron 2 into S2 cells in affecting the endogenous splicing or expression of the agglutinin or Drosophila agglutinin mRNA. So actually here, in this context, we could not actually express the full-length human agglutinin in flies because the intron 2 cannot be retained. So what we did is we forced the expression of this human cis-RNA by mutating the 5' splice site. of this intron 2 so that it cannot be spliced properly in the S2 cells. And we transfect the entire full-length human AGLU1 RNA in flies, as shown here. And with this, we also do not see that it has any activity or impact on the splicing or gene expression of the Drosophila endogenous AGLU1 transcript. So next we ask, how could this be the case? So we think that could it be the fly AGLU1 protein is not compatible with the human cis-RNA? So what we have tried is that we tried in vitro phase separation assay by incubating human intron 2 with fly AGLU1 protein. And indeed, we saw that if we incubate human intron 2 and fly AGLU1 protein, we do not see any increase in the droplet number, and also we do not see in the increase in the droplet size as well. As a positive control, we incubate Drosophila intron 2 with Drosophila AGLU1 protein, and we do see that the Drosophila intron 2 can induce phase separation by increasing the droplet count of Drosophila AGLU1 protein and also the size of the Drosophila condensate. So it seems like the human cis-RNA cannot actually bind properly to the Drosophila AGLU1 to induce phase separation. And that could be a reason why the human AGLU1 cis-RNA do not have any activity in the fly cells. So because of time, actually, I will not go through all the data that we have. And if you are interested, you can go to the paper that he recently published in EMBO reports. I would just like to bring you all to a summary that we have in this study. As shown previously, we think that the human AGLU1 cis-RNA is formed by intron 2 retention, and this actually draws the AGLU1 protein away from the locus, and this actually sequesters the AGLU1 protein in the nucleus speckles. However, in the fly, what we have seen is that actually, the fly cis-RNA is produced in a slightly different way. Instead of retention of intron 2, it is produced by premature cleavage of intron 2. And this actually occurs in a different mechanism whereby it involves U1 SNRP. And with this production of the fly cis-RNA, it instead draws AGLU1 protein to the nuclear, to the transcription loci, whereby inhibiting splicing further. That actually explains why fly and human AGLU1 cis-RNA have different function. Although they regulate splicing, human AGLU1 cis-RNA promotes splicing, whereby fly cis-RNA represses splicing. So I would like to end here by acknowledging people who have done the work, especially Chris, is the one who actually did 100% of the work that I've presented today, and also acknowledge investigators and scientists and colleagues that have shared their reagents with us. And here is actually TLL. If you are coming to Singapore, do feel free to visit us. Thank you very much. Appreciate to take any question.
Thanks, Jun Wei. That was really exciting. So there's 3 questions in the chat. So if we go in order, from Ines, how do you know the function is not coming from an element in the DNA instead of the cis-RNA? Have you tried to change the order of the nucleotides in the UCE instead of deleting it?
I think the experiments we have done is we did ASO, which knocked down the RNA specifically. So I assume that the ASO will not interfere with the DNA because we are not making any aberration or genetic mutations at the DNA level. So we think that this the effect is mediated at the RNA level. And we have not actually changed the nucleotide. I think that is a very interesting question. Yeah, so far what we have done is just remove that, and we have not actually tried to manipulate the UCE. Thank you for the suggestion.
Next question from Lisa. Might non-coding RNA alternative splice variants like this not be an exception, but more a common theme for protein coding genes?
Oh yeah, I think that's something that I mean, from a more biased view, I hope this could be like a really general phenomenon, and that is actually what we actually, I think I won't be very surprised if this could be a more general phenomenon than what we thought. Yeah.
So next question from Daniel. The definition of cis-RNAs is independent from biogenesis and function. Would you consider intronic mRNA, microRNAs and snow RNAs to be cis-RNAs?
Yeah, I think this is a question that I often get as well. I think it's also an important question, and I think in this cis-RNA, if you realize the field is not very big, so I think when Joe actually tried so-called coin this term, he doesn't actually try to say that this is like a class. I mean, in a sense that it has all the same function, or what. It's just like a name to describe a phenomenon. So with microRNA, actually, I believe all of us know, I mean, this microRNA has specific length and way of biogenesis, which is actually more definitive as a microRNA. So at this point of time, cis-RNA is more meant to be something like a phenomenon that is something that is a stable RNA. But of course, as we understand more about how the biogenesis or function of cis-RNA, the question is, could we actually find some common themes or maybe variations in processing of different subgroups of stable introns? And we can call them differently. But at this point of time, we are just calling them cis-RNA in general. So the definition of cis-RNA is intronic sequence, and they have to be stable.
So there's one more question from Matthew about, does the RGLU1 as cis-RNA target other alternative splicing events in other genes? And can the cis-RNA from the mini gene affect the splicing of the endogenous RGLU1 gene?
Okay. I think this is a very interesting question. So far, we have only tested the autoregulatory feedback loop between the RGLU1 cis-RNA in regulating its own alternative splicing. We haven't really tested how it affects alternative splicing of other genes, but we know that the RGLU1 protein itself can regulate alternative splicing of many other downstream genes because that is a protein function, and we know this RGLU1 cis-RNA can affect expression of the protein and in turn so affects splicing events of other genes. So at this point of time, we haven't really looked deeply into this. And can the cis-RNA from mini gene? Yes, I think when we overexpress this mini gene cis-RNA intron 2, it can actually affect the splicing of the endogenous RGLU1 gene.
Next question is from Stephen. The Bartel lab has suggested that cis-RNAs in yeast sequester splicing proteins to inhibit splicing under saturating conditions. Have you seen anything similar in flies?
Yes, I think we were very excited when we saw Bartel's lab, and actually there's another in the same issue of Nature. There's another lab that report the presence of stable introns as well in yeast. And what they have shown is that during starvation condition, there's an general increase in stable intron in yeast, and this actually affects splicing of ribosomal protein, if I remember correctly. And this actually helped the yeast survive during this stressful starvation condition. And whether we see this in fly, actually we did something like that in the fly ovaries, and we do see after if we starve the flies in the ovaries, many of the cis-RNA that we look at actually all go up. So now it's actually the time to look at more closely what are the individual function or collective function of this cis-RNA in regulating so-called oogenesis, because it might be done by some different mechanism, but we think that stable introns could be a more general phenomenon on how cells combat or respond during stressful condition.
So the last question from Jean Lawrence, are long introns disproportionately represented, more prevalent in cis introns?
Okay, long introns, that's an interesting question. To be honest, we actually, in my lab, we haven't really done that in flies, but I think in Xenopus or Joe's lab, they have done this so-called this bioinformatic analysis of different lengths of introns whether they are more prone to producing cis-RNA or not in the frogs. And they did see some so-called length bias of cis-RNA producing introns. Yes, so actually they are actually usually more introns that are shorter, if I'm not wrong. Yeah. So but we do not know whether this we don't see this happening in longer introns is due to technical artifacts, because sometimes the longer RNA get cleaved easier. so that we don't sequence is longer introns. But I think based on what they have found is that they have a bias for shorter introns, maybe around 300 to 400 nucleotide.
Okay. So I think that maybe because we do apologize that we started late, so maybe we can close the session because there's no more questions. So thank you very much to Anthony and Jun Wei again for sharing their work with us, and to everybody for really lively and engaging session. And I guess we'll see everybody in 2 weeks at the next seminar. Thank you.
Thank you very much.
Thank you.